In this video, I will describe two processes mediated by the plant hormone auxin. First is phototropism, so essentially plants grow towards a source of light. And we'll also describe gravitropism. So here we can see a potted plant has fallen over. Presumably it was growing up at the time, but due to a process called gravitropism, the shoot or the stem is going to grow up against gravity and the root is going to grow down with gravity. And so let's start with phototropism. And we're going to zoom in to cells within the stem in particular. Okay. Just as a reminder, plant cells have both a cell wall and a plasma membrane. Inside the cell wall, there are a series of organelles, but most relevant to this discussion are the vacuoles. The vacuoles contain water and they can generate turgor pressure, so essentially outward force on the plant cell, and essentially that is kind of pushing in all directions but limited by the cell wall. The cell wall here, right, is surrounding the entire cell. Right, we can see that over here as well. The pink line represents the cell wall, whereas the blue line or purple line represents the phospholipid bilayer, the plasma membrane. Here I have exaggerated the cell wall to show that there are a series of polysaccharides that are forming a network. Right, here we can see the largest and very pronounced polysaccharides called cellulose microfibrils. Okay. And they're kind of like woven together here, and they are secured with cross-linked fibers, and there are a series of these cross-linked fibers, but here I have just shown xyloglycans. So they are securing the position of these cellulose microfibrils. And so even as the vacuole takes on more and more water, there is no expansion of the cell wall because these fibers are held together so tightly. Okay. Usually in a plant, auxin, a hormone, the most common form of auxin is IAA. Auxin is synthesized in the tip of the shoot, in the apical meristem, and essentially it is going to descend down through the plant through a series of channels. Here on the apical surface of the cell, we can see one auxin channel called Aux1 or Auxin 1. And here, auxin, here the form IAA, is able to enter the cells from more or less the top of the cell. And then the auxin is passed down through exporter proteins called PIN. And there are a series of PIN molecules, but here just generically I write PIN. Okay, so this ultimately produces a flow of auxin from the tip of the shoot down through the plant. Now in the stem, auxin is going to stimulate growth, or at least expansion of the cells, particularly of the stem. Other cells promote growth in the lateral direction, so the branching, but auxin is going to promote growth in the stem of the plant. Okay, so that's normal. If light is kind of shining on the plant, roughly the same in all directions. However, if there is a light source from one direction only, as we know, the plant is going to grow towards it. And so what's going to happen in that situation is that light, particularly blue wavelengths of light, are going to activate a light-sensitive protein within the plant cell called phototropin. Okay, so this is going to change the shape of the molecule and therefore activate it. This active phototropin is going to do a lot of different things, but one that is very relevant for this discussion is that it translocates PIN exporters into the lateral wall, in fact, into the shady side, of the plasma membrane. Okay, so now instead of auxin just flowing down the cell or down the plant, now auxin is going to be pushed away from the light source. Okay, so here we can see auxin is going to go into the next cell over and into the next cell over yet. And so this over here is the shady side of the plant whereas the light side of the plant is over here with the translocated pin exporters. Okay, so auxin is going to have a lot of different effects on this particular cell, ultimately leading to the process of phototropism. The first one I want to mention is that auxin can bind to surface receptors. Here we see ABP1, auxin binding protein 1. And in response to this auxin binding to its receptor, there are going to be potassium channels activated or inserted into the plasma membrane, therefore leading to an influx of potassium ions. Water is going to follow this solute essentially via aquaporins and enter into the vacuoles. Now, this additional water in the cell is going to increase the turgor pressure. Okay, so how much pressure is being put onto the plasma membrane and ultimately onto the cell wall. Here we can see two representative cellulose microfibrils, which are held together by these xyloglycans. And so even though there's more water in the cell and therefore more turgor pressure, the cell is not yet able to expand. And that's where this other function of auxin comes in. Auxin can be imported via those same AUX1, and usually it would be up here, but just for demonstration, the auxin is coming across to the shady side of the plant. It enters into the shadiest cells, and therefore it's in the highest concentration, the shadiest cells. And through a series of different types of mechanisms, auxin is going to activate, right, through transcription, translation, and phosphorylation sometimes of different proteins. And this is going to activate, ultimately, a proton pump. And so hydrogen ions are going to be pumped from the cytoplasm into the apoplast. And so the space in between the cell wall and the cell membrane. This additional hydrogen is going to do a couple different things. First of all, it brings the pH down and therefore loosens up the connections between these cell wall fibers. Furthermore, hydrogen ions are going to activate a protein called expansin. And as the name suggests, expansin is going to allow the cell membrane to expand. That is, it's going to loosen up these connections here between the xyloglycans and the cellulose microfibrils and ultimately allow this, for example, cellulose molecule here to move. And so the turgor pressure over here is pushing on the cell wall, and now these cell wall fibers are no longer as securely connected to one another. And so turgor pressure is going to push the top of the cell up, and with it, push these microfibrils apart. Okay, so now we have this kind of weakened cell wall on this side, but also a longer cell. Okay, so if this light source continues to be over here for any extended period of time, auxin is also able to increase the synthesis of more proteins and ultimately more polysaccharides to further strengthen this wall after it has been expanded. Right, so that's kind of permanently or more permanently curving this stem towards the light source. Okay, so ultimately, auxin has led to the development of longer cells, not more cells at this point, just longer cells on the shady side than on the light side. And so these longer cells are going to ultimately push the stem towards the light source so that more can receive the light and therefore photosynthesize. And so this, again, is the process of phototropism. Ultimately, auxin usually goes from the tip of the plant and descends down the stem. But in response to light sensed by phototropin, these pin exporter molecules are going to be translocated or at least inserted into the membrane on the shady side of the cell, ultimately pushing auxin to the shady side. Auxin is going to stimulate the lengthening of these cells. Okay. So that is phototropism. Gravitropism also uses auxin and also is going to translocate pin, this time in response to gravity. Okay. So in this discussion, we are going to look at both the root and the shoot, that is the underground portion and the above ground portion of the plant. They both function a little bit differently because these guys need to go with gravity and the shoots need to go against gravity. Okay. So auxin is generated quite a lot up here in the apical meristem of the plant. It descends down through the plant, both through these transporter proteins as well as through the vascular system of the plant. Okay, so down here, we can see auxin is descending down into the root of the plant. And so here it travels through the vascular system. Ultimately, it's going to get down to the tip of the root. Here's the root cap. And it is going to be diverted back up. Okay, so that's usually how it works when the root is already growing down, right? When the plant is oriented with gravity already. Okay, so in order to do that, in order to redirect the auxin, pin exporters must be in the lateral sides of the plants within the tip of the root, which allows the auxin to be diverted back up. The reason for the positioning of these pin proteins is the presence of what are called statoliths. Okay, these are heavier molecules. They descend down to the bottom of cells with gravity, and in the root, they are going to stimulate the insertion of pin into the lateral walls. Okay. When the root is on its side, as is the case here, so the pot has fallen over, the roots need to grow down, but they're not growing down yet. So once again, we see auxin is descending down into the root. It gets to those cells within the tip of the root, and this time the statoliths aren't on this side of the cell, they are on this side of the cell. And that is ultimately going to redirect all of the auxin to this side. Right? So instead of some going in this direction and some going in that direction, now all of the pin exporters are on this side of the cell, and they are going to divert auxin down to the bottom of the root, bottom with gravity. Okay. In the root, unlike in the shoot, auxin is going to suppress growth. Okay. So these cells over here are going to remain relatively small, and over here, where there is virtually no auxin whatsoever, the growth is not suppressed, or at least the expansion is not suppressed, and therefore these cells on the top of the root are going to get longer. And so just like we saw with phototropism, the different sized cells on this side versus this side is ultimately going to lead the root to grow down. So over here, longer cells, over here, shorter cells, and this redirects the growth with gravity. Okay. In the shoot, right, so again, this is in the stem of the plant, not in the root. Here we have auxin being synthesized, right, in the apical meristem of the shoot, just like we talked about in phototropism. Usually, as we know, auxin is going to descend down the stem via these transport mechanisms as well as the vascular system. When the plant falls over, or when there is lateral growth, when there's vertical orientation here, horizontal orientation, pardon me, the statoliths are going to descend to the bottom of the cells. Once again, this is going to translocate pin exporters to the bottom of the cell. Okay, so now we see auxin, instead of just going down in this direction, it is going to go down with gravity as well. And so on this side of the stem, there's going to be a lot more auxin than there is up here. And so as we know, in the shoot, auxin promotes cell expansion. And so these cells down here with the most auxin, these little orange guys here, there's going to be more cell expansion down here than there is up here, which makes this side grow a lot faster than this side, and therefore the stem is going to grow up. Okay. So once again, this is gravitropism. When the plant is oriented horizontally, in the root, statoliths are going to redirect auxin distribution to the bottom of the root, which is going to inhibit growth and on the bottom and allow growth on the top, therefore inducing positive gravitropism. In the shoot, we experience negative gravitropism, so statoliths again descend to the bottom of the cell. This relocates pin to the bottom of the cells, so auxin is distributed to the bottom once again. This time auxin is going to stimulate growth on the bottom side, therefore promoting negative gravitropism or growth away from gravity. Once again, this was gravitropism, growing with or against gravity, and phototropism, growing towards the light. Both of these processes are regulated by the hormone auxin. Thank you.